So far, we've talked about inheritance of genes that are on chromosomes that are not sex chromosomes. And so now we're going to look at sex linkage and what is the inheritance pattern of genes that are carried on the X chromosome, because they don't necessarily follow exactly the inheritance patterns of other genes are on other chromosomes, which we call autosomes. Just a reminder of the chromosome composition of sexually reproducing organisms. So we're using human beings as an example. So in human beings, we have 23 pairs of chromosomes altogether. I'm showing you the chromosome lineup of a female in here and of a male here. So the first 22 pairs of chromosomes are called autosomes. So if we were going to draw a line, these are all autosomal chromosomes. They're autosomal. Okay. We also have our sex chromosomes. So in a male, it would be an X and a Y chromosome. The male determining gene is on the Y chromosome. For a female, it's going to be 2 X chromosomes. So then when we write the inheritance patterns of genes that are on the sex chromosomes, we either remember the naming convention. For a female, we write the X chromosomes and then we write that naming convention that we have for a gene as a superscript on the symbols. So let's say if we're looking at the gene for color blindness, normal is big C, and then color blindness is little c. So this big C stands for color. Okay, so that would be a heterozygous in a female. For a male, males only carry 1 X chromosome. The color blindness gene sits on the X chromosome. So this male only has 1 X. So it could be either big C or it could be little c. And obviously for female, you could have 2 big C's or you could have 2 little c's. You can have all these combinations. Genes that sit on the Y chromosome, well, obviously since only males have a Y chromosome, they're only going to express that gene, show that gene. So then the superscript goes on the Y chromosome. And we don't really run into many situations where the gene is on the Y chromosome. But let's just say some gene, I'm just going to call it gene A. Then we would be writing it as a superscript on the Y chromosome. Okay. Now, the fact that these genes are on sex chromosomes, their inheritance pattern shows sex dependency. And this is what we call X linkage. So just as a review, humans have 23 pairs of homologous chromosomes. 22 pairs of them are what we call autosomes, and 2 sex chromosomes, either X or Y. XX is female, XY is male. X and Y, as you have noticed in those images, they're not exactly the same size, so they're not homologous over their entire length, but there's enough homology between X and Y that allows them to line up together on the metaphase plate. So the first incidence of sex linkage was identified by Dr. Morgan, who studied genetics in flies. And the phenotype his lab was looking at was the color of eyes. Most flies in nature have a red eye, and that happens to be the dominant allele for red eye color. And some of them are they have white eyes, so they don't have red eye color. And I want to emphasize here that just because something is dominant doesn't mean it's more abundant. So don't mistake a dominant allele for abundance or strength or whatever. All a dominant allele means is that it can be expressed both in a homozygous dominant individual and in a heterozygous individual. That's all it means. Okay, so when Dr. Morgan's lab was studying the inheritance pattern of eye color, he noticed that there is sex dependency in terms of the ratios that he observed. So it depends on which sex he was looking at. So the cross that was made was a male fly with red eyes. So obviously the male has the dominant allele. And then the female was a homozygous recessive. So the alleles that are made by this male are here, lined up here, and then alleles that are made from the female are right up here. So both alleles are the same. Now, when you cross them, two X's will give you females. So both females, not both females, all the females from this cross always had red eyes, and all the males from this cross had white eyes. So scientists noticed that when they compared the sex of the fly and the color of eye, it really depends on the sex of the fly which color eye they had. And that was when X-linked genes and the fact that some genes are inherited depending on the sex of an individual was discovered. So human sex-linked disorders affect mostly males. So why is that? Well, first of all, many sex-linked human disorders are recessive. So why do we see them in mostly males? Well, because males receive a single X-linked X chromosome. So if you think about it, let's talk about in terms of hemophilia. If we say normal hemophilia is big H, and a hemophilic patient would have the little h allele. So a female could have a number of different genotypes, right? So a female inheriting a hemophilia allele from her parents could either have this combination or it could have this combination depending on the genotype of her parents. So females have a 50% chance of getting away with getting a disease but carrying the disease gene. But a male really does not have another X chromosome to rely upon for a 50% chance of survival. So if a male inherits the recessive allele, which is disease causing, it is going to get it 100%. So that's why conditions like hemophilia and color blindness and muscular dystrophy, you see it more often in males than you do females because of just the way the sex chromosomes that are present in males. Females can be carriers. This is called the carrier. Females can be carriers but not be sick, but males don't have an option to be carrier. So, studying human genetics using pedigrees. Now, with flies and plants and any other or model system that geneticists use, scientists have the freedom to set up crosses as many as they want, and usually for example, with a pea plant or flies. Those are great model systems because the generation time is short, and then there's many babies that are made at each generation. So that allows geneticists to do statistics. It's easy to do that. But with human beings, you really can't do that. You can't set up crosses at will to study human genetics. So all scientists are left with is what they have in human families and different generations. So instead of trying to rely on statistics, scientists try to kind of make sense of inheritance pattern of diseases. So that's why they create a pedigree analysis. So in a pedigree, a square is male and a circle is a female. Now in this image, they are shading normal and healthy, let's say, because we're looking at alkaptonuria. This is a disease. It's a recessive trait. They're showing them with different colors, but mostly in most of the pedigrees that you see, the affected individuals get a shading. So this would be someone who is affected. So you shade its symbol that is related to the person who is diseased. Obviously, you don't kind of go outside the line, but I don't have a lot of control. So this one would be a female that is affected by the disease, but a normal male would get a clear symbol. So it's a normal female. Okay, so an important thing to recognize with studying pedigrees is that you cannot rely on Mendelian ratios to predict mode of inheritance. So if we give you a problem like this and we ask you whether what kind of inheritance pattern there is, you don't go take a, let's say, look at these parents and say, oh, they have a 2-to-1 ratio, it means whatever. You really can't do that because you don't have enough samples, you don't have enough data points to be able to do that kind of statistical analysis. So what you have to do is rely on the behavior of genes throughout generations. Now, one of those behaviors is that recessive traits tend to skip a generation, so you can easily identify them. So what do we mean by skipping a generation? It means a case like here. So these two parents, they do not, neither of them have the disease. But all of a sudden in their children, you will see one of the children expresses the disease. So that means that the normal, the healthy version of that gene is dominant, because you really can't get a child that is sick without the parents of the child to be carriers. So therefore, if you're looking for clues as to the type of inheritance of a particular disease, try to see if you can find instances where you see the symbol, the person who carries the disease skips a generation. So when we say skips a generation, we don't mean the entire generation. Just find parents that don't express the disease, but all of a sudden they have children that do. So again, heterozygous individuals are called carriers for recessive traits. So these individuals, this mother and father, they are carriers of the allele, but they don't express it. Genotypes cannot always be deciphered for sure for those expressing a dominant phenotype. And we saw that before when we talked about the test cross. So for example, for this individual and this individual, we really don't know exactly what their genotype is, because there could be both big A and big A little a possible. So is in here. Sometimes there's more than one possible mode of inheritance. And that's because, again, coming back to not having data. So you really, the best way to deal with problems that involve pedigrees is to go with the process of elimination. Some tips for solving pedigrees. So especially when you try to see if you can eliminate X linkage, sex linkage. So if we give you a pedigree, for example, like this, and I'm going to solve this for a minute, within a minute for you, and we ask what is the inheritance pattern of the gene? Is it on the Y chromosome? Is it on the autosome? Is it dominant or recessive on the X chromosome? Go through the process of elimination to get you close to the final answer. So if the disease is due to a Y-linked gene, and since only males have a Y chromosome, only sons or only males would carry the disease. So if you see a pedigree where also females carry the disease, then that can't be, excuse me, a Y-linked gene. So only men would be disease carrier. That doesn't mean that all men are going to be disease carriers. It means that only you should see symbols that are shaded for males. Okay, so you could have males in the pedigree that look like this and look like this, but all the female symbols should be clear. You should not get any females with, it's supposed to be a circle. All the females, none of the females should have shading. All the females would be clear. So what about X-linked recessive gene? So one of the best ways that I've found to address this is to find an affected female. So let's just imagine that we have a female, let's say, with hemophilia, just for example. Okay, so this female, hemophilia is recessive. So if a female has hemophilia, they would have to be homozygous dominant. Now, let's say they marry someone, and we really don't really matter what the X chromosome of the husband is. Okay, let's just say, for this case, we're gonna have big H. Okay, so sons only inherit their Y chromosome from their dad. They don't inherit an X chromosome. So therefore, any phenotype then that the sons of this marriage show, it's because of the X chromosome that they inherited from their mom. So the only chance that males have in this case, if mother is hemophiliac, it would be hemophilia. This boy is out of luck. Okay? So look for affected females. Look for females that look like this. Okay? And then ask, when they marry somebody, what do their sons look like? If the disease is due to an X-linked recessive gene, the son should also be shaded, no matter how many sons they have. Okay? So this usually is a sign of X-linked recessive disorder. It's usually a sign, okay? But what you want to do is looking for exceptions, looking for conditions that would violate that. So if you see something like this, then it is not an X-linked recessive gene. So look for clues in the pedigree that would violate this rule. What about for X-linked dominant? Well, again, if the gene is due to a dominant allele, let's say, for example, dwarfism, okay? So dwarfism, it's the name of the disease, I've forgotten. So dwarfism is dominant, and normal individual is little d, okay? So what happens? How do you figure out whether the gene, the disease is due to a X-linked dominant allele, okay? So what you do, again, you look for a homozygous recessive female. So look for a disease-free female. So this would be her genotype. Her symbol would be like this, no shading, okay? Now, if this person, let's say just for argument's sake, marries a disease-free individual, or even marries a somebody who has a the dwarf syndrome. That's really not a disease, it's just a syndrome, because they can live full lives and accomplish everything the rest of us do. Okay, so then again, we're going to look at males. So males only get their Y chromosome from their dad. They get their X chromosome from their mom. So what is the only choice to have? The only choice they have is for them to be disease-free. So if you see something like this, this is a disease-free female, and let's say they marry someone. Let's just, for argument's sake, that they do have the disease. So the husband is this one. Okay. So if this is X-linked dominant, all the sons should be disease-free. But if you see a situation where one of the sons has the disease, then it is not X-linked dominant. So again, we want to look for patterns of inheritance that violate this rule so we can rule it out. So let's look at it in the context of a real problem. So examine the pedigree. Square represents male and circles represents female. Filled in squares represent the inheritance of a disease. Which of the following could be the mode of inheritance of this disease? So when you're given this problem, the easiest way always is to go through process of elimination, and the ones that are easiest to rule out is this one, disorder on Y chromosome. Can we find females that have the disease? Yes, we do. Since we can find females that have the disease, then it cannot be on the Y chromosome because females don't have a Y chromosome. So we rule this out. Now, recessive disorder on the X chromosome. We say we're going to look for a female with disease. So let's find one. So we have a female with disease. Let's look at her sons. All of her sons have the disease. But does that mean that we should stop looking? No. We look at all the female that have the disease. So here's another female with a disease. Her sons should all have the disease, but they don't. So that violates that rule. This cannot be an X-linked chromosome. Here's another female. Her son doesn't have it. Okay? Dominant disorder on X chromosome. Remember, we're going to look for a disease-free female. All her sons should be disease-free. So on this generation, we don't have one, but here we have this female. She is disease-free. All of her sons should be disease-free, but they're not. So that violates that rule. So we cross this again. So the only thing that is left is dominant disorder on autosome. That's the correct answer. Okay?